start recording. Going. Go with these buttons.
Oh, you are talking.
Some will edit. Some will cut it out.
It won't take time to cut it out. Because I need to put this on Zoom.
Today?
Yes.
Now?
Yeah.
Some can cut their parts out.
Okay.
Let me cut you before you start talking. Remove that thing of some out. The model, WhatsApp. Which Windows are you using?
10.
Windows 10.
Yeah?
You can start recording.
Welcome once again. We are going to look at immune system, part 3. And here I want to focus mainly on the specific defenses or the adaptive immune system. So the adaptive immunity is the third line of defense, and it is also called acquired because we get it post-birth. We get it as we move along with life, as we get exposed to various antigens. The production of specific antibodies is mainly done by the B and the T lymphocytes, and this occurs in the third line of defense. And it is also in response to an encounter with a foreign molecule. The foreign molecule is the antigen. The adaptive immune system is a functional system that recognizes specific foreign substances and acts to immobilize, neutralize, or destroy foreign substances. It also amplifies inflammatory response and activates the complement system. The adaptive immune system is antigen specific. It is systemic and also has memory. And we have already looked at this aspect. As I told you earlier, when you get exposed to a specific antigen, this adaptive immunity is going to act specifically and it will release antibodies against this antigen, and then it will store these for future use so that when you get infected again, your body will immediately sense and release these antibodies that were stored. So it has mainly two separate but overlapping arms. We have the humoral or antibody mediated, that is the B cell immunity, and the cellular or the cell mediated or T cell immunity. So let's briefly look at antigens. We said antigens are the foreign substances that come into our bodies, that the body senses and releases antibodies against them. So these are the ultimate targets of all immune responses. Antigens are large, complex molecules that are not normally found in the body. They are non-self substances. You can call them large complex molecules not normally found in the body. You can define that way. But you can also define it as a molecule that is capable of inducing an immune response in the host organism. And sometimes antigens are part of the host itself. For example, in an autoimmune disease, you find that the body starts attacking its own self. It fails to recognize the some cells as self. So once it recognizes these cells as non-self, then it will automatically release antibodies against these cells that are part of the body. So the antigens are targeted by antibodies. Let's look at some forms of antigens. We have complete antigens. And the complete antigens have important functional properties, such as immunogenicity. This is the ability to stimulate proliferation of specific lymphocytes and antibody production. And then reactivity, the ability to react with the products of the activated lymphocytes and the antibodies released in response to them. So these are some characteristics of complete antigens. They have immunogenicity and they also have reactivity. So some of the complete antigens include the foreign proteins, nucleic acids from the viruses that get into our bodies, then some lipids, and then large polysaccharides. We also have incomplete antigens. These ones cannot by themselves promote antibody formation, but they can do so when conjugated to a protein. So they are mainly called haptens. Most haptens are small organic molecules of low molecular weight. That's peptides, nucleotides, and many other hormones. And they are also not immunogenic as opposed to the complete. They do not stimulate a response. And then they are reactive when attached to protein carriers. Some examples of haptens, for example, poison ivy, some detergents, and cosmetics. So haptens, if they link up with the body's proteins, the adaptive immune system may recognize them as foreign and mount a harmful attack. And this is bringing us to the aspect of allergies. When something is not harmful to the body, but the body will sense it as harmful and release antibodies against it, and mounting a harmful attack, that is an allergy. Types of antigens. So antigens can be categorized into broad classes based on their origin. And so many molecules can function as an antigen in the body. And there is always a considerable diversity even within these categories. And these are the main classes of antigens that are involved in immune system activation. We have exogenous antigens and endogenous antigens. Those are the more specific types of antigens. And remember, we have also talked about the complete and incomplete antigens based on their ability to respond to antibodies. So the exogenous antigens enter the body from the outside, for example, through inhalation, when you breathe in a foreign substance, maybe foreign gases that are harmful to the body, carbon monoxide. Then ingestion by eating, when you eat some food substance that has an antigen that is not good for the body. And then through injection. The exogenous antigens are the most common kinds of antigens, and they include pollen, and then foods that may cause allergies, and then molecular components of bacteria and other pathogens that could cause an infection. We also have the endogenous antigens. These ones are generated within previously normal cells, and they occur as a result of normal cell metabolism or because of viral or intracellular bacterial infection. And these both change the cells from the inside in order to reproduce. The fragments are then presented on the surface of the infected cells in the complex with the major histocompatibility complex, that is class 1 molecules. Remember, we said that MHC is the one that binds onto this antigen. It binds onto the healthy cells so that they can be recognized as healthy. And so that when the macrophages or neutrophils come to attack, they will recognize only the cells that do not have the MHC and then destroy them. Cells that don't have MHC are harmful, and they are the pathogens. The ones with MHC are the healthy ones. So you find that sometimes our normal cells may lose their ability to maintain their MHC, and so the body will recognize them as foreign and destroy them. We also have autoantigens. Autoantigens are normal, and they are safe proteins, and are also complex of proteins. Nucleic acid that is attacked by the host immune system, for example, and it causes an autoimmune disease. Under normal circumstances, these antigens should not be the target of the immune system. But because of mainly genetic and environmental factors, the normal immunological tolerance for such an antigen gets lost. The body loses the ability to tolerate such antigens, and then now it starts attacking them as foreign substances. We also have tumor antigens or neoantigens. These antigens are presented by MHC1 or MHC2 molecules on the surface of tumor cells. And these antigens result from a tumor-specific mutation during malignant transformation of normal cells into cancer cells. So we find that many tumors have developed ways to evade antigen recognition and immune system killing. That's why they keep thriving. We also have the native antigens. This is an antigen that is not yet processed by an APC to smaller parts. The T cells cannot bind to these antigens, but they require that they be digested and processed by the APCs, whereas the B cells can be activated by the native ones without prior processing. So these are the main specific types of antigens: the autoantigens, the exogenous antigens, the endogenous antigens, tumor antigens, and the native antigens. So let's briefly look at antigenic determinants. Certain parts of an entire antigen are immunogenic. The antibodies and the activated lymphocytes may bind to these antigenic determinants. And we have some of these naturally occurring antigens that have specific antigenic determinants. These determinants mobilize several different lymphocytes and form different kinds of antibodies against the antigens. So this is the antigenic determinants. They have different shapes. And these antigenic determinants are specific to the specific antibodies. The shape will determine which antibodies they will bind to. If we are having these round shapes, then they are going to be able to bind to a curve-shaped antibody. This is antibody B. And then the one that is triangular is going to be able to bind to antibody A. And then we have this other shape. So the shape is determines the kind of antibody that this antigen will bind to. So let's have some specific characteristics of the specific immunity. One is specificity, the recognition of the foreign organism by the specific immune cells, and the activation of these immune cells to produce a specific response, for example, antibodies. That is what makes them specific. They respond or release specific antibodies to specific antigens. And this is why we are looking at the different determinants of antigenic substances. Then also memory. The lymphocytes are programmed to recall their first encounter with an antigen, and this will help them to respond rapidly to subsequent encounters. So let's look at self-antigens, the MHC proteins. Our cells in the body are dotted with protein molecules. These are the self-antigens, and they are not antigenic to us, but are strongly antigenic to others. And this brings us to the aspect of transplant rejection. You find that if somebody needs a kidney transplant, they are going to have to look for someone with compatible kidney. Otherwise, these kidney cells will react against the next person to whom it has been transferred. So these antigenic substances are specific to individuals. They are self-antigens, so that when they are transferred to another person, they will react negatively. So one of the types of these self-antigens is the one we've been talking about, MHC. We've been hearing the word MHC more commonly since. So MHC is the major histocompatibility complex protein, and it is marked as a self-cell. So we have two classes of MHC proteins. That's class 1 MHC, found on virtually all body cells, and class 2 MHC proteins found on certain cells in the immune response. Lymphocytes. Immature lymphocytes are released from the bone marrow, and they are essentially identical. Whether a lymphocyte will mature into a B cell or a T cell will depend on where in the body it becomes. We have the immunocompetent lymphocytes. These are the B cells that are mature in the bone marrow, and the T cells that mature in the thymus. So these ones, they mature, and then once they mature, they will stay in a specific location. The B cells will be in the bone marrow and the T cells in the thymus. So the B cells become immune competent and self-tolerant in the bone marrow. And some self-reactive B cells are inactivated while others are killed. And then some other B cells undergo receptor editing, where there is a rearrangement of their receptors. Then the T cells, we already said they mature within the thymus under negative and positive selection pressures. So negative selection means that it eliminates the T cells that are strongly anti-self. And then positive selection implies that it selects T cells with a weak response to self-antigens. So this becomes both immune competent and self-tolerant. Remember that the loss of self-tolerance causes autoimmunity. So immunocompetent B or T cells, they display a unique type of receptor that responds to a distinct antigen and become immune competent before they encounter antigens that they may later attack. And they are exported to the secondary lymphoid tissue where encounters with antigens will occur. And then after they'll mature into the fully functional antigen activated cells once they bind with their recognized antigen. And we have to know that it is the genes, not antigens, that determine which foreign substances our immune system will recognize and resist. Like earlier on, we saw when the cytokines are going to be released, the gene that releases the cytokines is first activated, and after which the cytokines will be released from the gene. So it is the genes, not antigens, that determine which foreign substances our body will recognize and resist. So this picture is showing us the sites of T and B lymphocytes. The site of lymphocyte origin is in the bone marrow. And then the site of development of immunocompetence as B or T cells primarily occurs in the primary lymphoid organs. So the primary lymphoid organs, as you can see, they are also inside the bone marrow. And then now, the one in green is the site of antigen challenge and final differentiation to the activated B and T cells. So, as you can see, in the red bone marrow, we have immature lymphocytes. And these immature lymphocytes will migrate to the thymus and the bone marrow, and then they will differentiate into the T lymphocytes and to the B lymphocytes. That occurs in the thymus, we have the T lymphocytes, and in the bone marrow, we have the B lymphocytes retained. So, number one, the lymphocytes are destined to become T cells, and they are able to migrate to the thymus, and then they develop immune competence there. Then the B cells will develop immune competence in the red bone marrow. And then once they leave the thymus or the bone marrow as naive immune competent cells, these lymphocytes will seed the lymph nodes, the spleen and other lymphoid tissues where the antigen challenge will take place. And the antigen challenge involves the final differentiation of these cells into activated B cells and T cells. So the mature antigen activated, the immune competent lymphocytes will circulate continuously in the bloodstream and the lymph and spread the lymphoid organs of the body. So once they become activated, they are able to now start roaming around, looking out for different pathogens to destroy. Antibody-mediated immunity. This is also called the humoral immunity. So this one involves the antigen antibody interactions in the body, and it is able to neutralize, eliminate, and destroy foreign proteins. The main function of the B cells is to become sensitized to a specific protein, which is an antigen, and also to produce antibodies directed specifically against that specific protein. So the cells involved in the antibody-mediated immunity, abbreviated as AMI. So they are also called antigen-presenting cells, or APC. Earlier on I mentioned the word APC. So APC means antigen-presenting cells. So some of the antigen-presenting cells we have are the B cells, the macrophages, dendritic cells, and the T lymphocytes. All these cells work with the B cells to start and complete the antigen-antibody interactions. And for optimal AMI, the entire immune system must function adequately. So all these do not work alone. They work as a group. They communicate within themselves to cause the effective function. Dendritic cells. These are antigen-presenting cells, also known as accessory cells of the mammalian immune system. Their main function is to process antigen materials and present it on the cell surface to the cells, the T cells of the immune system. They are also able to act as messengers between the innate and the adaptive immune systems. So the dendritic cells can be present all over tissues that are intact with the external environment. For example, the skin. Here we find a lot of specialized dendritic cell types called the Langerhans cells. And then also in the inner lining of the nose, the lungs, the stomach, and intestines. And they can also be found in an immature state in the blood. So once they become activated, they migrate to the lymph nodes where they interact with the T cells and the B cells. And then they'll be able to initiate and shape the adaptive immune response. And at a certain stage of the development, they will grow branched projections. And the dendrites, these branch projections are called dendrites that give the cell its name. So briefly, you will go and write about antigen processing and presentation. Task yourself, research and find out how this process of antigen processing and presentation takes place. So thank you very much. This is all we had for this class.